The information in this podcast is not intended to be used as the primary basis of investment decisions. Any forward-looking statements are not guarantees of future performance, and actual results may differ or other factors may change.
Welcome to Making Sense, the podcast for anyone interested in diving into the investing and startup space. Hosted by us, Emma and Luke.
Hey everyone, Emma and I are interns at Red Ventures, an investment and advisory firm based in Vancouver. From interviews to startup advice, we will take you on our journey as we learn more about the finance industry. Our guest today is Dr. Mads Daugaard. Mads is the co-founder, president, and chief scientific officer at biotech company Rakovina Therapeutics. Founded in 2020, Rakavina is striving to revolutionize cancer treatment and extend the lives of cancer patients. Not yet in any human clinical trials, they are developing medicines that selectively target and kill cancer cells using leading-edge science that we'll discuss today with Mads. The therapeutics company has 3 research programs, the KT-2000, 3000, and 4000, all of which are in the preclinical stage of drug development. Rakovina is currently being traded on the TSX Venture Exchange under the symbol RKV for 16 cents a share. This is my first time on the Making Sense podcast, but I know we've been lucky to speak to Mads in the past. And Emma, how'd that go?
Yeah, so I previously sat down with Mads back in November 2021, and we talked about Rakovina and their research at the time. In that episode, we were more focused on Rakovina's drug candidates that they were developing through the use of university partnerships with the University of British Columbia. Since then, I'm sure the company has had a lot of new innovation.
And today's focus is on the 2 conferences that Rakovina has been invited to speak at in the coming months.
On April 8th in New Orleans, he is attending the American Association for Cancer Research, or AACR, annual meeting, which is the largest cancer research conference. Definitely very exciting for Mads and Rakovina. Following that, he's sharing an abstract highlighting Rakovina's KT-3000 series at the AACR's special conference on sarcomas cancer in Montreal. And with that being said, let's get right into today's episode. Hi, Mads. How are you doing today?
I'm doing very well. How are you, Emma?
Yes, me as well. So thank you for joining us on the second podcast that we're doing together. If you wouldn't mind just starting us off and introducing all of our listeners a little bit about yourself.
Absolutely. My name is Mads Daugaard, and I am the president and CSO of Rakovina Therapeutics. Rakovina Therapeutics develops next generation DNA repair inhibitors for cancer treatment.
Could you tell us a little bit more about your role specifically in the company?
Yes, my role is to oversee the research and development for Rakovina Therapeutics. All that work is being conducted at the University of British Columbia, where I am also a professor, and I am overseeing the work on spot and in the laboratories of the university.
So what does this work on spot really entail then?
Well, it entails that our R&D research team that is engaged in the development work for the compounds in the company are working in my laboratories under my oversight at the University of British Columbia. And this very tight connection to academia is actually a pretty huge benefit for the company, because it gives us access to all the state-of-the-art technology and equipment that is available in a university setting through core facilities and other research infrastructure. If we did not have this opportunity, we would have to either set it up ourselves or contract the work with partners.
Interesting. Before we just dive too deep in this, I know Rakovina has 3 different series that you guys are working on, and I was wondering, just to set some foundation, if you could go over each of the individual series. What cancers are they addressing? And yeah, just a sort of quick summary about those 3 projects that Rakovina has going on.
Yes, absolutely. So as you say, we have 3 series that we are working on. We have the 2000 series, the 3000 series, and the 4000 series. And each series are designed to have certain added capabilities to what is already out there in terms of DNA repair inhibitors. In the 2000 series, we have specifically aimed to improve how these drugs can cross from blood circulation into the nervous system or the brain. It's something that becomes very important in the context of metastatic disease, where you often see spread of a primary tumor to the brain. And because the human body has this barrier between blood and brain for various reasons, that is normally serving as a very functional and very good barrier. In the context of treatment, it actually is an obstacle. So we want to make these drugs easier penetrable into the brain. And for the 3000 series, we have combined 2 capabilities that are normally found in 2 different drugs, but we have combined it into one single molecule. And the 2 capabilities is, on one hand, the ability to directly inhibit DNA repair. And on the other hand, we can inhibit some of the other pathways and capabilities that cells need to facilitate DNA repair. So you are actually attacking the whole DNA repair machinery from 2 different angles in the same molecule. And that's an attractive way to do it, because if you are working with a single molecule instead of a combination between 2 different molecules, then you can eliminate a lot of clinical complication, usually surrounded treatment regimes where you take 2 different drugs and combine them in a treatment. And that is overlapping toxicity that you often see when you put 2 drugs into patients. And also all drugs behave differently. So how long they stay in the blood and how much exposure you actually get to the tumor will be different from drug to drug. But if you have it in the same molecule, you know what you're dealing with, and it's much easier to fine tune.
Very interesting. I just want to switch the topic a little bit here. I think in the coming days, you might be off on a flight to a conference, and I know you have another conference coming up. So I was hoping you could just explain those 2 conferences that you're attending, tell us a little bit about them, and why it's important for Rakovina to attend these conferences.
Yes, Rakovina has a very clear strategy on how we want to present data and publish data that is generated on our series and make that information available to our investors. And that is through these peer-reviewed scientific conferences. We like that vehicle because our research and development activities are peer-reviewed by colleagues in the field when they are accepted for presentation into those conferences. And that kind of validates our work by our peers. And also it's a good way for us to get feedback during those conferences from our peers that actually have expertise in the field. For the two upcoming conferences that Rakovina will participate in, they are actually both American Association for Cancer Research conferences, so what we call AACR. The annual meeting is just about to kick off in New Orleans, and Rakovina will be presenting data at that meeting related to the 4000 series that we are yet to talk about. And just a little bit later in May, we will present data at the AACR Sarcoma conference with the KT3000 series that we think will have a special role for sarcoma types of tumors.
Well, I'm excited about the special type of role, but before we dive into that, do you think you could just give a little bit, going back, could you give a little bit of an overview of this 4000 series and how you're going to be presenting it at this first AACR conference?
Yes, the 4000 series, as with the 3000 series, is actually also a combination drug that can do two things at the same time. So it can inhibit DNA repair on one hand. On the other hand, it can actually induce DNA damage that needs to be repaired. So the idea behind this type of molecule is that you actually create the mayhem that needs to be repaired in the tumor in the same molecule. And we think that is an attractive strategy moving forward, because there has been quite a few indications from both preclinical work, but also combination studies in the clinic, where you have combined these two treatments sequentially with some benefit.
Interesting. If you don't mind explaining just a little bit more, if you're inhibiting the repair while simultaneously inducing the damage, how are these cells going to become healthy again if they're not able to, like, I know these are the sick cells that can't regenerate, but is it affecting the healthy cells as well?
We are not trying to make the tumor cells healthy.
Okay.
We are trying to kill them.
Okay.
So what happens is that if a tumor cell cannot repair its DNA, then it will go into basically a cellular suicide, a programmed cell death that we often call apoptosis in our field. And that's what a program in all cells, where they can be either triggered by other cells, or they can choose from internal mechanisms inside a single cell to go into a suicide mode. Then the question becomes, well, will this affect the cells around the tumor that are healthy? And it will to some extent. But what is unique about cancer cells is that they proliferate a lot more than our normal cells. And so it means that the DNA damage you induce in this molecule will mainly target only proliferating cells. So you have a selectivity built in to the treatment, where specifically dividing cells and proliferating cells will be sensitive to this. And that's the whole concept behind this molecule. And in principle, the concept behind standard chemotherapy as well.
That's very interesting. Are these molecules that you're developing, are they able to treat different types of tumors in all different types of cancers, or are they tailored towards specific types of cancers?
Yeah, that's a very good question. Usually, until now, the DNA repair inhibitors has been deployed mainly in cancers where you find mutations or defects in the endogenous DNA repair system. But if you combine these, different ways of inhibiting the entire process of DNA repair, as we are trying to do with the 3000 series, then we think that you will be able to overcome that requirement and potentially deploy these treatments beyond where the current DNA repair treatments are deployed. And it will be in solid tumors. So when I say solid tumors, I'm referring to basically all cancers that start out as a solid tumor, but has the potential to metastasize.
Are there other companies who are using a similar two-part approach then, or is this just specific to Rakavina?
To my knowledge, we are the first company that takes this approach, and our series of molecules will be first in class when they eventually go into the clinic.
Now you mentioned, I guess the first Rakavina is the first to do this. Is there anything that compares to like a similar therapy, and if so, has that been successful, or what other science have other companies used that's similar in respect to what Rakavina is doing?
Yeah, so DNA repair inhibition has been around for quite a few years by now. There are actually four treatments or 4 different drugs that are approved by the FDA and also Health Canada for treatment of these types of tumors that have defects in their DNA repair machinery. There's been a lot of, incensement and tests over the past years for trying to see if you can deploy those inhibitors outside that space where you have mutations and defects in your DNA repair machinery. That has so far not been very successful. And that is why most of these companies that produce these types of drugs here, they are testing combination treatments where they combine it with another drug in the treatment regime. So, there are indications out there that certain combinations of capabilities will have a benefit for many cancer patients, but just how to fine-tune that and how to obtain that result, that's a whole different question. And what Rakovina is doing is one way to address this.
Very interesting, and I guess that's why it's so important to present these findings at these conferences. You said you were going to 2 of these recent in the upcoming future, near future. What is the difference between these 2 conferences?
The difference is mainly the focus. The first conference here, the annual meeting for AACR meeting, is a very big meeting that encompasses all types of cancers, all types of drugs, and all types of basic preclinical research as well as clinical research. The second conference is very specifically targeted the type of cancer that we call sarcoma. So it's a type of cancer that occurs in our connective tissue. And many of the sarcomas are cancers that are childhood cancers. For example, Ewing sarcoma, which is one of the sarcoma types that we are very interested in for various reasons. That is a cancer that is mainly found in children and in young adults. And if you have a spread of Ewing sarcoma, your probability of survival is around 5 to 10% over the next 5 years. So there's a strong clinical need in that specific indication to find new treatment opportunities.
Mm-hmm. And this was the conference you said you were especially excited about going into this next little while. Do you mind telling me a little bit more about this abstract that you're going to be presenting at the Sarcoma conference?
Yes, we are going to present data on a few selective compounds from the KT3000 series that has the ability to inhibit a DNA repair enzyme called PARP. And at the same time, it can inhibit an enzyme that is called HDAC, or histone deacetylase. And inhibiting those two enzymes at the same time has a quite profound effect on Ewing sarcoma cells. That is interesting, of course, because of that, but it's also interesting because the PARP inhibitors have actually been tried in Ewing sarcoma patients before. And it was not like they were toxic. It was not like they didn't work at all, but they didn't work as good as you had anticipated in that trial. And that is probably because Ewing sarcoma are usually not very mutated in their DNA repair systems. So they have mostly intact DNA repair. So it's a type of cancer where you would need something else coming in to inhibit that system at the same time. And that's where the combination with HDAC inhibitors makes sense. Also because there's plenty of scientific literature and preclinical work available out there that actually highlights the potential of HDAC inhibitors in Ewing sarcoma. So both kind of moieties that we are combining into a single drug formulation has been tested in Ewing sarcoma one by one. That's why we are excited about this.
What exactly are PARP inhibitors and HDAC inhibitors?
Yes, so PARP inhibitors inhibit an enzyme called PARP1 and PARP2. There are actually two